so we can finish. There's one more amino acid cycle, tryptophan. Okay, tryptophan is the source of NAD, okay, and NADP. Okay, so that's one thing. Tryptophan can also be used to make melatonin. It could also be used to make serotonin. Serotonin is made first and it's converted to melatonin. In this case, you can use deoxygenase. In this area, you can use dehydrogenases, but it really doesn't matter. I mean, nobody cares for this. Multiple steps involved. The only thing is tryptophan deficiency will lead to these problems. Decrease in serotonin production, depending where, and NAD, NADPH, and melatonin. Melatonin is not needed for what? Wake sleep cycle, right? Wake sleep cycle. Very good. Okay. Serotonin is needed for what? So serotonin is the same thing as, you can also call it 5-hydroxytryptophan. It's just simply hydroxylation. Let's just add OH. Okay. To tryptophan, it becomes serotonin. So serotonin is the same thing as 5-hydroxytryptophan. At position 5 of tryptophan ring, you add OH group, it becomes serotonin. Serotonin has many functions. So it's needed for platelet aggregation. Platelet aggregation. So it can cause thrombosis. Risk of thrombosis will be increased. Serotonin sometimes also, for example, in pain, you know, like bee stings and stuff like that, serotonin can give you a sensation of pain. It's also needed for act as a neurotransmitter. Okay, so again, in this case, it makes you, gives you some concentration, focus, wakefulness, arousal. Okay, so lack of serotonin can, for example, lead to depression-like symptoms. Okay, and remember, we will talk about SSRI, right? So in this case, we have to know that selective serotonin reuptake inhibitors affect serotonin concentration in the synaptic cleft somehow. How? It increases it, which means serotonin decrease is found in depression, major cause of depression. Okay, serotonin causes vasoconstriction as well. Vasoconstriction. Okay, so there's multiple receptors for serotonin. So 1, 2, 3, 4, 5, depending what we're talking about, they're found in different parts of location. 1 primarily in the brain, 2 platelets. Okay, 3, 4, and 5 in the brain and GI. So, but what you need to know is, number 1, it is needed for wakefulness and arousal, because melatonin and serotonin has same thing. Okay, platelet aggregation, vasoconstriction. See, platelet aggregation and vasoconstriction should be easier for you to understand. Why? The purpose of platelet aggregation is to prevent bleeding, correct? So vasoconstriction also prevents bleeding. So they both occurs at the same time. So serotonin induces both of them. Serotonin can be made by platelets themselves. So platelets will have the 5-hydroxylase enzyme, tryptophan 5-hydroxylase. So this enzyme hydroxylates tryptophan at position 5. Platelets have them. So does other cells. Okay. Deoxygenase. Okay, so removing oxygen from tryptophan, so you get NAD, NADPH. So okay, deoxygenase. I'll put oxygen negative. Okay. So just know for those things. At the end of the day, there is nothing more to it. Here you don't have to have multiple cycles and stuff. The metabolism is straightforward. It's the product. By far the most important product of this pathway is serotonin, of tryptophan. Serotonin makes you feel awake. Melatonin makes you feel asleep. It's a completely different thing. Okay. Melatonin, for example, you know when they say you eat turkey, turkey is rich in tryptophan, so you get more melatonin, you start getting drowsy and sleepy. So melatonin gives you drowsiness, serotonin gives you wakefulness. Okay. These are nicotinic amide dinucleotide. That's what it stands for. Nicotinic amide dinucleotide. and this one is nicotinic amide dinucleotide phosphate. Okay, these are coenzyme cofactors. Okay, now you can have niacin in the same thing. Niacin. You heard of niacin? B3. Yeah, niacin and vitamin B3. Okay. So tryptophan problems can lead to pellagra, for example, a disease known as pellagra. Okay. And this disease, again, this is malabsorption of, let's say, tryptophan, or technically deficiency of vitamin B3. Okay, so B3 deficiency. B3 deficiency is known as pellagra. It has 3 D's. Do you remember that? Diarrhea, dementia, and what else? Dermatitis. Okay. So you can have pellagra-like symptoms when you have B3 deficiency alone. But if you have tryptophan malabsorption, it's known as Hartnupp's disease. Hartnupp's disease. It's in your notes, okay, and it's in your PowerPoints everywhere else. Hartnupp's, H-A-R-T, and then Nupp. Hartnupp's disease is when you are unable to absorb tryptophan in the GI, in jejunum. Huh? H-A Hart, H-A-R-T, and then N-E-P, Hartnupp. N-A-P? U-P. Yeah.
What is that, B3?
Yeah, vitamin B3. So that's one thing. And you can have another disease known as?
Dr. Munir,
Yeah.
what's the cause of the not absorption of the B3?
Yeah, so do you know that there are some amino acids they're known as aromatic, and neutral amino acids? Okay, neutral, aromatic. What are those amino acids? One of them is tryptophan, and what's another one? Okay, phenylalanine. Okay. Huh? No, tyrosine has OH. It's not neutral. Okay. Phenylalanine is neutral and aromatic. You can have methionine, which is only neutral, but it's not aromatic. Okay. So malabsorption of these amino acids. All of these amino acids has a unique transporter. It's known as neutral amino acid transporter. So in the GI you have cells. They're known as enterocytes, correct? Enterocytes will have a receptor, a channel that transports tryptophan in association with sodium. If you have decreased concentration of this channel or complete deficiency, this will lead to tryptophan deficiency or tryptophan malabsorption or neutral amino acid malabsorption. Other amino acids, because they can be synthesized and so forth, tryptophan is essential, so it becomes a big issue. So mostly Hartnup disease leads to tryptophan deficiency. And as a result, you will have deficiency of what? Niacin, serotonin, and melatonin. So it would affect in that sense. Again, Hartnup disease is mostly diarrhea. Diarrhea. Okay? It will lead to diarrhea and some sort of depression as well. But Hartnup disease will have pellagra-like symptoms. You will have them. Again, diarrhea, dementia. Dementia is what? Global deficit of the brain, aphasia, apraxia, and what else? Agnosia, and what else? Amnesia, and what else? One more A. Huh? Anomia. Yeah, you cannot name stuff. Okay. And dermatitis, because squamous cells requires B3. Squamous cell regeneration. So dry skin, it's scaly dry skin. In that sense, dermatitis, H. Okay. Alright, so that's for tryptophan. So when it comes to tryptophan, please remember pellagra and Hartnup disease. But now let's go to one more thing. Do you remember the granulocyte we talked about? Did we talk about granulocytes? neutrophils. Let's make them brighter. Why do we do this? There you go. Okay. There were some granulocytes who had granules within them. And if the granule stains blue, what do you call those cells? Basophils. You know, just for the sake of simplicity, I'll make a huge granule. Okay? Although the granule should be what? Smaller. And how many nuclei should they have? Multiple nuclei. So let's just, for the sake of understanding so you don't forget, granulocytes should have multi-lobe nuclei and many granules. Let's say this is one granule. You know what? I'm not going to do this. I'm going to do this. Because you guys know what it looks like, right? Okay. So these granulocytes, known as basophil, have an enzyme known as histidine decarboxylase. So let's put the amino acid histidine, and this can be converted to histamine. Histamine. Okay? And there's an enzyme. By decarboxylation, look, whenever we put amine, that means there's less oxygen, there's more amide or nitrogen. So histamine, you remove the carbon dioxide, okay, from histidine, it becomes what? Histamine. The enzyme should be called what? Histidine decarboxylase. Okay, we don't care if you know that. The idea is, as long as you know histamine, the source of histamine is what? Histidine, which is an amino acid. Okay? Whereas histamine becomes sort of a local factor. It's not an amino acid anymore. It's a keto acid without, I mean, it's a amide group without the carboxylic group, so it's just a local factor. And this histamine is then packaged within these vesicles or granules, which stains blue. So this cell is known as basophil, as long as this cell is found where? In blood. But when it comes to tissue, it becomes what? Mast cells. It's the same cell. Within blood, it's known as basophil. Outside of blood vessel, it's called, in tissue, mast cells. Okay. Now, these cells can package histamines within these granules, and the source of histamine is what? Histidine. The only concern we have is, please remember, histidine can become histamine with the help of histidine decarboxylase. Decarboxylase. Okay. Now this is the important part, because we're talking about amino acid. This lecture is about amino acid. But now I would like to go further. These granules can attach to the surface and open up in this process known as degranulation. Degranulation. And they can release what? Histamine. So let's say histamine is released. Histamine can do few things. Number one, it causes bronchoconstriction. Bronchoconstriction. Let me tell you this. Histamine causes asthmatic attack. If you can memorize that, the rest is easy. If histamine causes asthmatic attack, and if asthma is difficulty breathing, would you have difficulty breathing if your airways are shut or they're open? Shut. So that means they should cause bronchoconstriction. Can we do in 6 minutes? I mean 6%? I don't know how long it takes.
Remember, I give you the warning.
You did? I'm like one of those people always go last minute things. I don't know.
You don't feel it unless you feel it.
There you go. You don't feel it unless you pain it. So bronchoconstriction should be automatically. You don't have to memorize that. Okay, so histamine causes bronchoconstriction. Histamine also increases secretion of fluid. Secretion. Now the secretion would be mucus, would be tears, and saliva. So it increases the secretion. And you see the asthmatic patient, when they go under attack, they're having trouble breathing, and they're tearing a nasal discharge and so forth. Okay. Okay. Histamine also causes vasodilation, and that's the bad part of it. Vasodilation, you know that if blood vessels dilate without the volume being changed. Look, blood volume is not changing in this case, right? You're not adding more fluid or removing more fluid. You're just causing vasodilation. If that happens without volume being changed, blood pressure will drop. And as a result, the heart rate should increase to compensate for that. And this is shock. So histamine can cause shock, anaphylactic shock. It can cause bronchoconstriction or asthma. Let's just put asthma here. And the one sign of asthma will be what? Wheezing, right? You see, I'm trying to switch my technique now. I don't want to just give you purely boring. I'm going to try to relate to diseases so it makes more sense so you can relate to it and get your interest going. Okay? Otherwise, I can just teach biochemistry purely. But by doing this, there's only one issue though. Please study hard. This way it makes more sense. You can get more interest. But at the same time, information increases and it becomes difficult. If you study today, you'll be fine. If you leave it for tomorrow, you're not fine. I'm telling you, if you ignore it today, there's no way you can recover. No way. It will take you a whole 2 weeks of just studying on your own to get back to this level. So that's why whatever you recover today, you go home, take your break, eat and relax, and go back immediately. After 2-3 hours of going home, go back. Don't leave it for tomorrow. Don't leave it for tomorrow. If you do, you're... Is there a mnemonic for that? If you do, you're doomed. You're doomed, right? Okay. Alright, so this is what it is, histamine. Now, we need to know further. This cell is basophil or mast cells. Okay? On the surface of mast cells, we have IgE receptor, correct? Let me do this. The surface of basophil and mast cells have IgE receptors. Do you remember that? Okay. IgE is not produced by these cells. It's produced by what? B cells. B cells. So B cells produce IgE, and then these cells have a receptor on them. Let's see what happens. There's an allergen, smoke particle, cold particles, or let's say dandruff, or let's say animal dandruff to be specific, pollens from trees, whatever. And they come there, they're circulating in your nasal cavities, or they're going in your airways and stuff, and you have these IgGs that are produced in response to that. So the IgG comes and attaches. It attaches to this. Now the IgG can come, let's say, attached to 2 of them at the same time. This thing can come, and who has receptor for this? Yeah, basophil. Basophil. That's right. I'm gonna talk like somebody who knows what he's talking about. That's right. Let's bring it down. Let's rotate it. And then put it back here. And now we got the product. And relax. And breathe. Okay, there you go. So, number 1. See, the pollen must attach to 2 IgGs at the same time. The IgGs can be already on the surface, or they can attach the pollen first and then come and attach. It doesn't matter. Okay, that's not important. So, I will tell you the steps of allergic reaction now. Okay? So, you know the function of histamine. We don't care for that anymore. Now, the idea is, how do basophil release histamine? We know how basophil can make histamine, correct? It will take histidine, decarboxylate it, and make it convert to histamine, and then put it into these vesicles and packages and store it. So basophil always have these stores of vesicles. They're granulocytes, right? We've seen them multiple times under microscope. You guys probably haven't, I do. So you see them, they have these granules full of histamine. They're just sitting there. But they need a signal. They need a signal for these granules to attach to the surface and open up and release histamine. And now we're going to talk about how do you cause degranulation. Okay, so you want to get up to this level. This is degranulation. Degranulation. Okay, number 1. Pollen, or let's say allergen. I mean any antigen that stimulates the IgGs in this case. Allergen. Yeah. IgE?
IgG.
No, this is IgE. Did I say IgG? I'm wrong if I said that. IgE. It's an antibody. Did I answer your question? Okay. I don't know what you asked me, honestly.
Yeah, I mean, we're talking about the IgE, whatever it is.
Okay.
This one's just IgE receptors on it, right?
Okay, so
Just the pollen, so.
yeah, let me start all over. Okay. So there's an allergen. Allergen in this case is an antigen that causes allergy. Okay, antigen is a broad term, but when you use the term allergen, it's a specific antigen that causes only allergies. It would not cause any other condition, only allergies. Is that okay? Allergen can be dandruff, animal dandruff. It could be smoke particles. It could be fumes from factories and stuff. All those things that contributes to asthma. They're all allergens. So it depends what you're allergic to. Huh? Milk. What about it? Anything can cause it. Yeah. So, but milk, the antigen from milk has to reach to that level. If it's digested in GI, no. Most likely allergies are caused by what? In this sense, in asthmatic allergies, by inhalation. But when we talk about reactions to food product, it's a different scenario. Yeah. Okay, so allergen. Now, first there should be allergen. Second thing, this allergen must bind to what? Allergen must bind. Very good. IgE. Now please say surface IgE, not free floating IgE. It's usually surface IgE. Okay. IgE. So we'll attach to IgE. Third thing, the allergen should cause, it should bind to two IgEs at the same time. Let's just put that. Third, it will cause IgE dimerization. Dimerization. Let me show you on a picture how this would be done. Just to kind of make sense. Let's say one IgE is attached on the surface here, the other one is attached there, and these two have to be attached or occupied by the same allergen, large one. Okay, so when this allergen attaches to two IgEs at the same time, it allows these IgGs to contract. It pulls the two IgEs. I keep saying IgGs, I don't know why. It pulls the two IgEs together. That's called dimerization. And let me do that, see if I can do that. Dimerization. It pulls the two IgEs together. This is known as IgE dimerization. But the IgE is already attached to the surface. Imagine this. If you have a balloon, you put two glue sticks, whatever, on top, and then you pull them. What happens to the balloon? It stretches. If there's something, bubbles attached on the surface, they can open up. So this dimerization leads to degranulation. And the fourth things would be degranulation. And of course, degranulation causes release of what? Histamine. And you know the rest of the story, right? If this is the first exposure, no big deal. Now, if the first exposure sensitizes the basophils so much that they start producing a lot more IgE receptors, that means even a small concentration of this allergen can activate degranulation of multiple basophils, and it will cause allergic reaction. Okay, at the end of the day, it is not the number of basophil, it is degranulation that causes allergic reaction.
The dimerization will cause like membrane stability where it's in rupture if it's too strong.
Yeah, so for example, if there's old IgE, basophils, yeah, of course. But that's why they're killed by apoptosis, so that means it's a protective mechanism, so you don't have a release. If it's necrosis, yeah.
Autoimmune reaction is different than...
Yeah, it has nothing to do with this. Autoimmune reaction is cytotoxic T cells and antibodies themselves.
It's not related to histamine.
No. Okay, we'll talk about that. So you gotta know this whole thing what I talked about today. No basophils and mast cells have stores of histamine known as granules. and these granules can rupture and open up and release histamine, and it's the histamine that will cause the problems. But of course, histamine is stored somewhere. Okay, so relate that to histidine and decarboxylase, and that should help us out. Okay, there were diseases you said? Maple syrup and cystinuria. Hyperammonia? Okay. Cystinuria, what does it mean? Yeah, too much cysteine where? In urine. That's all. A lot of cysteine in urine. We talked about phenylketonuria. Okay, we talked about alkaptonuria. Okay, ketones are usually, you know, they once they go in kidney, they're excreted. But there is something else. How many of you know of nephron? Nephron. I have to quickly go over it because, as I said, there's no such thing as isolated knowledge. There's no prerequisite in that sense. The only prerequisite you have for medical school is your undergrad. But no matter what order I do medical school, it doesn't matter. Okay, so we have some basic knowledge you gotta have. Nephron should look something like this. Should have the Bowman's capsule, proximal convoluted tubule, thick and thin. Let's just do this. And then thick, and then distal convoluted tubule, and collecting ducts. Okay, and then they should have thin loop of Henle. Okay, so you should have basic understanding of what is a nephron. If you don't like this color, in textbook this color is usually slightly, there you go. There you go. Now it's true nephron. Okay. Right? Yeah. Okay. Now, things are being filtered from a blood vessel known as the glomerulus. Red is blood vessels. So this will filter everything in here. They will filter, leak in here, and it will go in the nephron. Amino acids are also filtered. Amino acid. All amino acids are filtered. There's no amino acid that's not filtered, so let's ignore that. All 20 amino acids are filtered freely because the gaps are large enough to fit amino acid. But protein, never. Never. They're too large. But there's the thing, amino acids are very essential for us. Remember, nucleotide synthesis depends on what? Amino acid. Muscle synthesis depends on what? Amino acid. Protein synthesis. Enzyme synthesis. Your whole body depends on amino acids. It's a big deal. So we better not waste them. We work so hard. You guys went to medical school. Some of you actually studied hard. Some of you are still sleeping and coming late and so forth. But whatever. You work so hard to do what? To provide your body with amino acid. Honestly. It all comes to that. You know? Even when you watch TV, there's a group of amino acid that causes reaction in your brain and gives you happiness. I mean, no matter what you do, it all comes to what? All the hard work you do is just for this. So why would you want to waste it? Therefore, 99% of all amino acids filtered are reabsorbed immediately. Immediately. This region, where I have the arrow, is known as proximal convoluted tubules. And this area right here is distal convoluted tubule. Convoluted tubules. This is known as the loop of Henle. And this is the collecting duct. So look, it doesn't even reach, the amino acid doesn't even make it as far as the thick ascending loop. Especially thin ascending, and then ascending, and then distal, and blah blah. No. It's immediately reabsorbed. The purpose of kidney is not to filter amino acid, it is to get rid of toxins. But of course, if toxins are larger than amino acid, then of course amino acid will be automatically filtered. Okay? So we reabsorb them. The fact that you find amino acid in urine is not a good sign. You shouldn't. It's common sense. 99% in the proximal convoluted tubule. Some can be reabsorbed here, the other 1% along the nephron. That means all of it is reabsorbed. When you urinate, you should find 0 amino acids or very small concentration. You shouldn't waste amino acids in urine. It shouldn't be wasted. So when we talk about cystinuria, that means there's some problem. we're unable to absorb cysteine, for example. Okay, so we're unable to absorb cysteine. We cannot do this. That means cysteine accumulates within nephron. It's not being reabsorbed, but there is a transporter specific for cysteine. It's known as the CoLA transporter. CoLA transporter. So cysteinuria is CoLA transporter deficiency or defect. And remember, in the proximal convoluted tubule, amino acids are symported in association with sodium. Okay, do you understand what I just said? Okay. You cannot absorb amino acid without sodium. Symport, they both transport in the same direction. So, let me do this again. When we talk about reabsorption, what does reabsorption mean? Sometimes students will get confused with that term as well. Let's see, this is a blood vessel. This is a blood vessel. Okay. This blood vessel is along, so I mean in my diagram it's like that, but in reality, I'll do it quickly and then I'll erase it. In reality, this blood vessel goes along the tube like this, and it will give you tiny branches that loops around the nephron. Okay, this is the peritubular capillary. Okay, peritubular capillary. I can keep it, right? I wouldn't bother you. Would it bother you if I keep it? No? I did it already. Yes. So this one is peritubular capillary. Capillary are small blood vessels where exchange can take place. Arterial, no exchange. Arteries, no exchange. Only capillaries are capable of exchange of nutrition and waste product. So now reabsorption means we take it back from urine into blood. That's reabsorption. So I'll put an R here. Reabsorption, taking stuff, whatever we filtered from blood. Look, filtration is from blood into where? Into urine. Filtration is from blood into urine. Reabsorption is once you filter something, you get it back into blood. Okay, now CoLA transporter stands for C for cysteine, O for ornithine, L for lysine, A for arginine. It's in your book, so I'm not going to spell it for you. Okay, CoLA transporter, C for cysteine, O for ornithine, L for lysine, A for arginine. This single transporter is able to transport all of them. All of them. So there's a unique transporter found where? In the proximal convoluted tubules of kidneys. All kidneys. In the proximal convoluted tubules of kidneys, you have a transporter known as CoLA transporter. And one letter for each amino acid. If this transporter is defective, can you reabsorb cysteine, ornithine, lysine, arginine? Yeah, this will affect the urea cycle too, because ornithine was needed. Okay. So, but anyhow, that means ornithine, as long as you have ornithine in your diet, it's okay. So, ornithine is soluble in urine, lysine is soluble in urine, arginine is soluble in urine. These four are, guess what? They are basic amino acid, especially lysine and arginine. You know that, you have to memorize. Huh? Yeah. Okay, so these are basic amino acids. When you decrease the pH of urine, what happens to basic amino acids? They dissolve better. They dissolve better. But cysteine is acidic. Not naturally, but it could be acidic. How do I spell acidic? Okay, it's acidic. Therefore, if pH of urine decreases, cysteine will precipitate and form stones. Okay, when urine pH drops, cysteine will precipitate. If urine pH becomes acidic, cysteine will precipitate. Arginine, lysine, ornithine will dissolve better. So the problem is, it could be okay in people, but when they have this deficiency and suddenly their urine becomes acidic because of they took too much acid or something else, then you have precipitation of cysteine. It will crystallize. And crystals are sharp molecules, and they can hurt. and they can make kidney stones. So the person will be at risk of what? Kidney stones. Somebody had a question about kidney stone. What was your question, guys? Did you have it? Huh?
Kinds of stones.
Okay, so this will precipitate as cystine stones. Cystine stones. Now this is genetic, right? So children will have a risk of kidney stones. Okay, and the symptoms of kidney stones are: number one, pain, severe pain, colicky pain. Pain is colicky. What does colicky pain mean? You know when your colon is cramped and it hurts? Severe pain. Okay, so it's very severe pain. And they will have bleeding. What's the term for bleeding? Hematuria. Now, some will say hematuria, that's fine as well. Now, you have to be careful, because when we use the term bleeding, you want to see blood with your eyes. That's natural, right? So this can be microscopic or macroscopic. You might not be able to see any blood, but you do a test for it, hemoglobin test, you might find it. So regardless, both will indicate hematuria. And of course, nausea and vomiting. Maybe fever too, but no. These three are important for kidney stone. Okay, so look for cystinuria. It will always cause kidney stone problems. But upon acidification of urine in children, and these are amino acid stones, usually they're not so good. You cannot see them on CT scan. They're not so good. You might have to use ultrasound. Okay. They're non-pigmented stones. So that's for this. Okay. Now, how would you treat a patient with cystinuria stones? Lots of water, but also alkalinization of urine. If somebody said that, I didn't hear you, I apologize, but yeah, you're correct. Alkalinization of urine, because what causes stone precipitation? Acidification. So you have to reverse that alkalinization. It will dissolve the stones and it will pee it out.
What about lysine, arginine, ornithine?
They're not known to cause problems. Do you want it to? Okay.
Could you destroy rupture the crystals into smaller pieces, or would you want to do that?
Yes, I would want to do that.
Ultrasound.
Lithotripsy, yeah. I would want to do that. Now, the criteria for stones, see, I don't discuss it here because this is not kidneys yet. But the criteria is stones can be, if stones are greater than between 2 millimeters, okay, so less than 2 millimeters, you can actually use ultrasound. If it's less than 2 millimeters, you can just use hydration and pee it out. They're small enough. If they are greater than 2 millimeters, but less than, let's say, about 2 to 5 centimeters, then you can use what? Ultrasound to break them. Okay. If a stone is 5 centimeter or bigger, you have to do surgery because you can't break it. You have to remove it. Okay. Laparoscopic surgery, go through a ureter. If it's inside the kidney, it's difficult. In the kidney, you have to break it. But if it's in the ureter, you can approach it and get to it. Then yeah. Okay. Last thing, then we can go. Oh, we're finishing on time today. That's good. Even though we took time for hypoxia and all that stuff. That's good. And we should be done with the amino acids. So one more thing. There's something known as maple syrup urine disease. All I need to put is an A, will become Masood. Yeah, Masood. I think my cousin's name is Masood. I think so. I have step cousins, and I left Afghanistan 23 years ago, and I haven't gone back. So, and no communication, anyways. So, you know how it is, right? Okay. Okay. All right. Maple syrup urine disease. Is this disease common in Canada? They have a lot of maple syrup, right? Upstate New York, Massachusetts, yeah, Maine, and all those. Yeah, sure. Okay. Now the problem here is there are 3 amino acids that are important: valine, isoleucine, and leucine. Okay, 3 amino acids. Letter represents each of those. Valine, isoleucine, and leucine. These are known as branched chain amino acid. Branch chain amino acid. The chain of valine looks like what? Please say V.
Upside down.
There you go. I mean, you can flip the amino acid, it will be V. The chain of isoleucine looks like what? I. Of leucine? L. They're all methyl groups. They're all methyl. So all chains, for example, for valine, it is CH2 or CH and then CH3 and CH3. That's for valine. This is the side chain. For isoleucine is what? Okay. And CH and CH. Okay. For L, you can have, let me just ignore this one, just not confuse it. It's the same thing, but of course, I don't know if it's 4 or 3, but whatever, and then add one more. L, I mean, upside down L, sorry. Don't make confuse you. There you go. This is L, this is I, this is V. Yeah. Okay. What did I say? Huh? 4? Oh, the number? I don't care. I thought you said, I said instead of isoleucine, I said something different. Okay. So now, these are branching amino acids. So, valine is glucogenic. It's glucogenic and ketogenic. These two are ketogenic. So, isoleucine is converted to acetyl-CoA. Acetyl-CoA. All ketones are made of acetyl-CoA. All ketones are made of what? Acetyl-CoA. So any amino acid that becomes acetyl-CoA is known as what? Ketogenic. Valine can become both. But let's say it's converted to, for example, glucose or pyruvate. Then this is known as glucogenic. Okay? So if you want to metabolize these amino acids, of course, this is what happens. They're converted to pyruvate, acetyl-CoA, acetyl-CoA. Now these amino acids, especially valine, you know that valine is a huge amino acid when it comes to something known as gluconeogenesis. Let's talk about this. Gluconeogenesis. This guy named Neo had a lot of glucose because he was fighting left and right in that Matrix movie. Had a lot of sugar. Gluco means glucose. Neo means new. Genesis means formation, production. Excellent. So it simply means making new glucose from scratch. When you want to make new glucose, you have to have the source of glucose, not glucose itself, because it's not new. If you convert glycogen to glucose, it's not new glucose. The only way you can have gluconeogenesis, making new glucose, if you convert lipids or protein or nucleotides into glucose. Okay, so valine is amino acid. If you want to convert to glucose, it's known as gluconeogenesis. Okay, but first it has to be converted to pyruvate. I don't want to go too much into details. And pyruvate is then converted to glucose. Okay, too many steps. But anyhow, so now all these 3 amino acids are used when you have less energy, when you have less glucose in blood. So the signal for production of these, breakdown of these amino acids will be when you have hypoglycemia. Hypoglycemia. So when somebody has low blood glucose level, then of course these ones have to be used to restore blood glucose level. Otherwise you can go unconscious. You can have seizures, comas, and hallucination when people are hypoglycemic. Yeah.
Glucose is stored as glycogen in the body.
Liver and muscle, yeah.
Yeah. So why those are involved in the conversion?
Okay. Very good questions. You have it stored in the body in the form of glucose, known as glycogen, correct? All of you know that you can store glucose in your body as glycogen. One is in muscles and the other one is in liver. True? Liver glycogen is only used for restoring blood glucose level. Muscle is only used for muscle. Let's say you're hungry, you don't eat. And what happens to your liver glycogen?
breaking down.
It's depleted. It's broken down. Okay, you want to use that term, that's fine. It's depleted, it's done. You don't eat for 6 hours. So your liver glycogen is done with. Now what are you going to do? You have no other options but use something else to make glucose. So unless, now in your case you might be eating glucose 24/7, you never have this problem.
No,
I'm just kidding.
When you are sleeping,
There you go. When you're sleeping, you will be using all the glycogen. If it's gone. See, the thing is, people who have high metabolism, when they're asleep, especially people who dream a lot, what organ of the body uses 80% of your plasma glucose? Brain. So when you're actively dreaming, then glucose is being used. Glycogen is being broken down and depleted. And then in dreams, sometimes you want to fly, sometimes you want to kick, and your muscles are rigid or you're scared. Your muscles are using what? Again, energy, glucose. So now you have hypoglycemia. When you have hypoglycemia, you have to restore that somehow so your brain can get enough oxygen and energy. So therefore you have to convert either lipids or proteins to glucose. The biggest source of glucose in gluconeogenesis is amino acid. In valine and aspartic, 2 amino acid. Memorize those 2.
this is the preferable pathway to
Yeah.
restore the glucose from amino acid. So it starts with amino acid. What if we have amino acid deficiency?
If you have amino acid deficiency, good luck. We have a problem. Yeah. Now let's talk about, there are people who live in Sudan. And you talk about Kawashikor. These people, you see they have a big belly, but you can see their bones. They have protein deficiency. Not naturally, but from diet. Okay, so yeah. That means you're starving. So at that point you'll be breaking your bones, dissolving your muscle, dissolving your skin, everything eventually, so before you can survive. Huh? Yeah. Yeah. They have protein poor diet. Yeah. Okay. So now, in order to convert them, there's multiple steps, but I only care for one step. Valine has to be made to keto acid first. Keto acid. So let me see if I can break this area. Okay. Valine is made into, converted to keto acid, isoleucine to keto acid, and leucine to keto acid. And then they're converted to what? To pyruvate or acetyl-CoA. Keto acid. Alpha keto acid. Alpha keto acid. Alpha keto acid. Okay? They're converted to that. But guess what? Now, alpha keto acid should be further metabolized. But there's an enzyme known as alpha keto acid dehydrogenase. Alpha keto acid dehydrogenase. So, dehydrogenase. Alpha keto acid dehydrogenase. It's the same enzyme. Works for all 3 of them. And if this enzyme is deficient, it will lead to maple syrup urine disease. Okay? So it's alpha keto acid dehydrogenase deficiency that will lead to maple syrup urine disease. So it's a genetic disease. Genetic disease. Alpha keto acid dehydrogenase. Alpha keto acid dehydrogenase deficiency. Alpha keto acid dehydrogenase deficiency will lead to this disease. Okay? Okay. Now in those cases you will have increase in what? In valine, leucine, isoleucine, or keto acids. They will increase in blood and in urine. Okay? When people start peeing, their pee will be now, remember that, in this case it's not a reabsorption problem like in cystinuria. You just dump too much amino acid into urine because they're not being converted. If they're not being converted, they'll be dumped into urine, and urine flows. Whatever you can absorb, but keto acid are usually not so good in absorption, so they will accumulate in urine. And then urine will have the smell of maple syrup. And that's why it's called maple syrup urine disease. Now, symptoms. Okay, so the problem is, so what if I have keto acid in my urine? Who cares? Maybe when I pee, there will be a lot of ants and bacteria and other pathogens. They will feast on it. They will have a picnic. Good for them. How does it affect you? That's the problem. The problem is when you're hypoglycemic, you will develop symptoms because you don't have these amino acids. You cannot convert them to what? To glucose. So this will only become problem in starvation or in fasting state. When you fast, when you're hypoglycemic, that's when it will be a problem. Otherwise, it's asymptomatic. And of course, the symptoms will be of hypoglycemia anyways, right? It will be of hypoglycemia, the symptoms. Hypoglycemia. Low blood sugar level. Don't ignore my arrows. So just hypoglycemia. And this, when you're low in blood sugar level, you will feel confused. Okay, headaches, blurry vision. Let's be honest, when you are really hungry, how do you feel? Not good. Just focus on that, that's enough. When people are hungry, how do they feel? They're aggressive, they're cranky, they're in a bad mood. They cannot think straight. All of those things, so think of that. Okay? And that's it. When you test for urine, you will find alpha keto acids in urines. Okay? And they will smell like maple syrup. So that should be diagnostic. What do you do about it? Avoid valine, isoleucine, and leucine diet rich in those amino acids. Avoid them. So stay away from those amino acids in your diet if you can. Okay? Just eat more sugar. If you eat enough sugar, you don't have to make new one. We already have stored, so we're okay. Okay, there you go. Yeah. Are these questions related to this? I don't have to turn off the thing. Maybe people can benefit.
I have a question related.
Okay.
Okay, if you have vitamin B1 deficiency, you can also have the disease.
Vitamin B1?
Yes.
Vitamin B1 is huge issue. Now, vitamin B1, enzymes that require vitamin B1 are affected by it. Alpha keto acid dehydrogenase, a little bit, not so much. Mostly this affect pyruvate dehydrogenase and transketolase. Okay, those enzymes. And malodehydrogenase. Those are an alpha ketoglutarate dehydrogenase. Those are the enzymes affected by B1 deficiency. Okay.
Tyrosine